There are actually several factors in orthobiologics, and it varies a bit depending on which orthobiologic that it is. But the factors that tend to be pretty consistent amongst them, that enable all of them to some degree to aid in healing, are a wide variety of things, from cytokines to growth factors, some enzyme inhibitors, to a big group of structures called extracellular vesicle. And each orthobiologic potentially has a different makeup of these various factors. And they can come from, again, from quite a range of sources. Stem cells can be a source of them. Components of the blood can be a source of them. You can actually isolate them from the blood, including the platelets, which is the source of platelet-rich plasma, or PRP. And certainly birth tissues have all of these factors, and all of these different sources can be used as the source for these biologically active molecules or factors. My honest answer as to how they exactly do they work is, I don't think we really know. But it is possible to talk about each one of the literally hundreds of different factors that have been identified, and you can talk about each one as an individual and have some idea of what that one factor, that one molecule does based on its mechanism of action. But exactly what happens when you combine a whole group of these factors and they act in concert, it becomes exponentially more complicated. But in a nutshell, what these factors do is they tell cells that they need to do something that is conducive to the healing process, whether it's telling cells to divide, whether it's telling cells that you need to produce more blood vessels, a whole range of things that they're capable of signaling other cells to do something that leads to tissue healing. Sure. The cytokines they're small proteins. They're what are called non-structural proteins. They don't form the backbone for an organism as a support structure. They're just small proteins, usually less than 30,000 kilodaltons or 30 kilodaltons. And what they do is act as messengers between cells. In your prior question about how do they work, cytokines are messengers between cells. They're involved in every biologic process to some degree, from triggering the inflammatory process and healing damaged tissue to aiding the development of new blood cells and the formation of blood vessels. They're critical to every biologic process because they act as a signal that tell those cells to do something that's important to that body. Cell signaling is really, it's the communication between cells and cells in their environment, especially other cells in their environment. It's how cells send and receive and then process messages from other cells that function to influence their biological activity. So the basic mechanism of how it works is that a cell signaling molecule, like a cytokine, for example, or we'll talk later on about things like growth factors, those molecules will bind to a specific receptor on the surface membrane of a target cell. And upon binding to that receptor, there's a message transmitted into the cell, and a lot of the time that directs how that target cell expresses specific genes as a response to seeing that cell signaling messenger. And it's most of the time it's used for constructive purposes, but it can be used for destructive purposes as well. Growth factors are, again, they're generally non-structural proteins. They're messengers. And for a lot of people involved in research in this area, they consider them to be a subset of cytokines. And they're small, non-structural proteins that function to a huge extent as cell messengers. And the reason that they're called growth factors is that the name originally came from the fact that the first several of these factors to be studied had a positive influence on cell proliferation. They caused them to grow. Thus, they coined the term growth factors. And similar to cytokines and other cytokines, growth factors do stimulate cell proliferation, wound healing, and even cell differentiation and maturation. So they work to stimulate the growth of cells. What's really nice about Hilltop Bio is that they've done extensive proteomic analysis of their product, and that's really the only way to know what is in your specific product. And some of the many growth factors that this proteomic analysis has demonstrated to be in Hilltop Bio's products are several of the growth factors that are potent regulators of cell proliferation, cell migration, cell differentiation, and even survival, including things like vascular endothelial growth factor, or VEGF, platelet-derived growth factor, or PDGF, insulin-like growth factor, IGF, and epidermal growth factor, or EGF. Those are four really important regulators of cell growth and proliferation, which is a huge component of healing. Exosomes right now are a very hot topic in research in a wide range of different areas. So exosomes are one of two different types of extracellular vesicles. Extracellular vesicles are tiny membrane enclosed sacs. The other type of extracellular vesicle besides an exosome is called an ectosome. And these, both ectosomes and exosomes, are intracellular. You can think of them as intracellular messenger bags that carry cytokines that we talked about, growth factors that we talked about, and these exosomes even can carry messenger RNA. Obviously, that's the code that tells how to make a protein. They can carry all of those factors, those messengers, between cells. And because of the important role that they play in cell signaling, extracellular vesicles are really a hot topic of research right now in everything from orthobiologics, like we're talking about, to cancer research. And knowing how all of these cargo molecules that are packed into the little membrane enclosed sac, the messenger bag, if you will, knowing how each of these can influence cellular functions, it's really easy to see why studying exosomes is such an active area right now. All types of cells can generate extracellular vesicles. If we look at something like stem cell therapy, it's widely accepted now that the clinical benefits that are associated with stem cell therapy are actually a result of what's called the secretome that they release, which in large part are extracellular vesicles, exosomes and ectosomes. The benefit from stem cells is not that the cells themselves stick around and divide and differentiate. It's they deliver these messenger bags to the tissue. Now, if you look at platelet-rich plasma products, the contents of those are quite variable, based both on the animal, in this case the horse we'll talk about, that is donating the blood to be processed, as well as the processing method itself that's used. And really, without doing proteomic analysis of this product, you really don't know what, if any, extracellular vesicles or exosomes are in a given PRP product. So I'm not aware of any of the PRP products having undergone a rigorous proteomic analysis looking for exosomes. One thing that's nice is that if you do know that exosomes are in a product that you're using, it does allow you to be confident that those really important cell signaling messengers that are critical to growth and to healing are part of what you're delivering to your patient as part of your therapy. TIMPs or TIMPs, it's an acronym for tissue inhibitors of metalloproteinases. And the TIMPs are a group of endogenous molecules. They live just about everywhere in the body, and their function is to inhibit one of the classes of enzymes that break down what's called extracellular matrix, which is essentially it is the structure in which a lot of cells will live. It's especially important in things like articular cartilage, which is made up largely of water, extracellular matrix, and then the occasional cell, cartilage cell or chondrocyte. So these metalloproteinases break down that extracellular matrix, which affects the structural integrity of things like cartilage, and it makes it weaker. It's part of the degenerative process. So because the TIMPs inhibit this one class of enzymes, they are important regulators of the turnover of this matrix, of tissue remodeling, that in all tissue remodeling involves matrix of some type, as well as the cellular behavior associated with those. So the TIMPs play a significant role modulating cell growth and differentiation, and they play a pretty large role in angiogenesis, or the formation of new blood vessels, which is obviously a critical function in normal tissue remodeling as well as wound healing. And what's been revealed within the last several years is that, interestingly, some of these TIMPs even demonstrate cytokine-like behavior and may have some anti-inflammatory effects and dampen the development of inflammatory-related pain. So I think we'll see a lot more to come out in the literature in the relatively near future about TIMPs. I think we've just begun to touch the tip of the iceberg, so to speak, about what we know about TIMPs at this point. And I think what really makes it interesting is that I don't think we've identified yet synergies between different factors that are there, competition between factors that are there, and trying to come up with the quote-unquote perfect mix. It's a challenge. One thing, and it kind of is self-serving for me, is I think the more that you can learn about the various different products that are out there, the better equipped that you'll be to make really good decisions for each individual case that you're dealing with. I don't think, given the complexity that's involved, I don't think that you can say that, okay, I'm going to use this one product and I'm going to use it on everything and it's going to make everything better. That's not realistic. But the more that you learn, the more that I think you'll be able to customize your treatment to whatever the problem that you have. And don't forget, I think a lot of these products are complementary. Don't look at them as competitive. Look at them as complementary. There's a lot of really good attributes, and there's a lot of really good products out there. And don't be afraid to ask questions, whether it's of yourself or other people, about what's in them and how you can use those to best benefit your patients.